morning's session of Speciation Genomics. We're going to, for the first talk, hear from Andy Kautt about cichlids. And so, you might need to wait for the dawn, but there you go. Perfect timing.
Okay, thanks. So my name is Andy Kautt. I'm a postdoc with Axel Meyer at the University of Constance. Today I'm going to talk about a project in which we use population genomic data to test the evidence for sympatric speciation and its alternative in Nicaraguan Crater Lake cichlids. And this is currently in press in PLOS Genetics, so I'm happy to talk about this later on, but I also invite you to check out our paper. And to put this in a broader context, if you don't like the term sympatric speciation, you can think of it as the most extreme case of primary divergence with gene flow. And we're interested in this, first of all, because it addresses the fundamental question, really, if geographic isolation is necessary for speciation or not. And second of all, if we have a putative case of sympatric speciation, the hope would really be that it makes it easier to get at the stuff that matters. Yeah? The idea being that, of course, gene flow will homogenize the neutral parts of the genome, while divergent selection will have a really localized, demarcated pattern here that we can pick up. And there's some of this in the paper, but I'm not really going to talk about this. Instead, I'm going to focus on the demographic and phylogeographic aspects to really address, okay, is the assumption of sympatric speciation actually true? And Crater Lake cichlids have for long been considered, so to speak, prime examples of sympatric speciation. Although last year, a study cast some doubt on this in Cameroonian Crater Lake cichlids. Our system is in Nicaragua. You can see here it's located in Central America. And it not only harbors the two largest freshwater lakes in Central America, Lake Nicaragua and Managua here, but also a line of different crater lakes. And then fish have been colonized from the Great Lakes, or the Crater Lakes have been colonized from the Great Lakes here. And today I'm going to focus on those two crater lakes because there are several endemic species have been described. And the crater lakes are really remote and also small, so they offer no geographic barriers within the lakes. And so Lake Quiloa has so far been less well studied, but in Lake Apoyo, around 10 years ago, a group of people in the Meyer lab made a strong case for sympatric speciation. The main line of argument of evidence came actually from this, monophyly, the mitochondrial marker. See, this is the source population. There are two species here in the crater lakes. That time only two were described, now we have more. And not a single haplotype is shared, yeah? They all share this mutation. And this monophyly was also later supported by genome-wide nuclear markers. However, the initial study here was also criticized. And part of the criticism came from this, so this is basically genetic space, and you can see that one Crater Lake species here is a kind of intermediate position between the source and the other Crater Lake species, which we wouldn't really expect from sympatric speciation. And the authors of this criticism actually stated, okay, this intermediate position is incompatible with sympatric speciation. I think the statement is somewhat too strong, as I will argue later, but it hints at something really important, which is that monophyly alone is actually not sufficient to make a case for sympatric speciation. Well, why is that? We can think of several scenarios that could lead to monophyly or apparent monophyly. The first one, well, this is just really simple sympatric speciation after single colonizations. Imagine the fish here in the Great Lakes. They come into the crater lake, and then here in situ they diverge and speciate. So we expect a single colonization, and they should be, so this doesn't really work, equidistant. So genetically, they should be equidistant to the source population. There's a second scenario here, which so far has been, I think, underappreciated. Just one of sympatric speciation after a secondary wave of colonization. So imagine we have one colonization here, and then a second wave, and then so those bring in some alleles that then get incorporated into the, get incorporated into the gene pool of this crater population, and this may or may not facilitate speciation here in Sympatric. But we expect cell colonization, they should be again equidistant to the source and should show equal admixture proportions. And we can still think of this as sympatric speciation as population divergence itself actually happened in Sympatric. The alternative, which is not sympatric speciation, is one of secondary contact and integration. Imagine we have one colonization event, the population already starts to diverge and is already reproductively isolated partly from the secondary wave, and then through reinforcement or ecological displacement we get two cell species here. And here, you would expect them not to be equidistant to the source. Yeah, the second wave of colonizers should be more closely to the source population than the other one. And this is really the three main scenarios we wanted to test. Here you can see an overview of the focal species and sample sizes. So there's two sets of species, it's the same set of species in the Great Lakes. So do we have another? This is, okay, sorry, no. So this one and this one is the same species, and this one and this one. In Lake Quiloa we have 4 described species, in Lake Apoyo 6. Although some of them have been described just based on morphological data. We'll come back to that later. We have a decent population sample, 25 to 50 for most, and did individually level red sequencing. So the main axis of genetic variation looked like this. This is the first axis of the PCA superimposed on a geographic map. and all the lake populations form nicely distinct genetic clusters. Also here on this admixture plot, you can see this. However, it's also apparent that here actually we have two distinct clusters, one being slightly closer to the source population, which shows some admixture, and those correspond to exactly 2 out of the 4 described species here. So this could be a signal of secondary contact and introgression. However, if you look at a more likely number of clusters, the signal actually disappears. What you can also see now is that within the crater lakes we find, or also the other lakes, we find distinct genetic clusters. If we zoom into this in Lake Quiloa, we have evidence of voice support for 4 genetic clusters. We have 4 described species. There's a bunch of individuals that are somewhat intermediate, which resemble hybrids, and they correspond nicely to the 4 species. And the hybrids are not showing this, but they're also morphologically intermediate. In Lake Apoyo, the situation is a bit less clear. We have evidence for only 5 genetic clusters and 6 described species, and only in this one case here do we have a nice one-to-one correspondence. But for the remainder of the talk, I will just use those genetic clusters here as objectively defined taxonomic units. First, we're interested in evolutionary relationships, and the crater lake flocks have a very high support for monophyly. Yeah, here 100% crystal supported here. The x-axis, so this was done with tree mix based on the frequencies. The x-axis represents genetic drift, so basically a measure of effective population size. And coming back to this apparent signal of admixture here, individuals here belong to this and this species. And you can see the branch lengths are much shorter than in the lineage here. So I really think this is not a signal of admixture, but rather it's due to the fact that they have a larger effective population size, have retained more of the ancestral variation. You can also look at it another way if you look at the residual covariance matrix. This is a bit complex, let me just walk you through this. So given this tree topology, here's always the pairwise covariance is basically left and unexplained. Yeah, this is a heat map. Dark colors indicate there's something that doesn't really fit nicely in the tree because maybe there was some gene flow between species. And we do find evidence for this, but importantly only within the crater lakes, yeah, here and here. But among the different lakes, we don't find any evidence for this. There's no evidence for differential admixture. We also did formal tests in terms of F3 statistics, and none of them were significant. So the monophyly here, strongly supported by the evolutionary relationships within the crater lakes, was not very well resolved. Yeah, very low bootstrap support. To further investigate this, what we did is we used SNAP, which is designed for B allelic markers and uses the multispecies coalescent. And interestingly here, Lake Apoyo, see at this point here, there really was a burst of speciation. It doesn't really follow a nicely bifurcating pattern. And similarly in Lake Quiloa at this point, those 3 lineages seem to split almost simultaneously. This has a striking resemblance to the outcome of a theoretical model proposed 10 years ago by Dan Bolnick, where in symmetry after 400 generations, one population splits into 5 or 6 ecologically diverged species. So we really think Cichlids might be, to our knowledge, the first empirical example of this multi-species outcomes of sympatric speciation. And so far what I've shown you is our data is consistent with sympatric speciation, but now we really wanted to explicitly test the 3 different scenarios. I'm just illustrating here again, yeah? Sympatric speciation after single colonization, one after admixture, and secondary contact. The problem is F3 statistic and so on might not be very sensitive for this one, and they will probably not be able to distinguish between those 2 scenarios. And to test this, we built demographic models. So we performed coalescent simulations with FastSync-Pro2. The data was summarized in the side frequency spectrum. And here I started with very simple one population models in the source populations. We go in coalescent terms, we're going from the present to the past. In forward terms would be here from top to bottom. Yeah? And then we can see the evidence for population-less changes, exponential growth, and so on. And this spotted growth scenario was the most supported one. And then based on that, I added for each crater lake species, or built 2 population models. You can see here, so this would be the crater lake population that comes out. Then we can test for evidence for gene flow, exponential growth, and importantly for admixture events. So this would mean we have one colonization here, then a second wave colonization, which then the alleles get incorporated into the gene pool here, and then yeah, this is the crater lake population. And interestingly, really this model was the most supported one for all of the crater lake species. So coming back to our three main scenarios, there's really not much evidence for this one, yeah? And to test those 2, I framed them in full 5 population models. So those are represented here. Yeah, so really frame the evolutionary hypotheses in demographic models to test them. This is sympatric speciation of the mixture, one colonization event. We have an admixture event here, and only after that we have speciation in symmetry. The secondary contact model, we have one colonization here, then a secondary wave colonization, and then admixture between the 2 crater lake populations already. And then after that, further divergence speciation events. And those models are highly parameterized, and I really want to focus your attention on some of them. I don't have time to talk about all of this, but those are some key parameters concerning the colonization history. So here, and I should say this is for Lake Apoyo, our model, maximum estimates give us that around 1700 generations ago, Lake Polio was colonized by 260 individuals. It's then been growing, and then 900 generations ago, there was a mixture event of 4%, and after that, shortly after that, we have sympatric speciation. In the secondary context, the integration model, the first colonization event is very similar to this one, and then 500 generations after that, we have a secondary wave of colonization, again by a similar founder population. But then only 250 generations after the secondary wave of colonizers has been established and the population has been growing, around 90% of its gene pool would have been replaced by the resident population already, which I think is biologically a bit more unlikely to think of. And if we look at the likelihoods, this model is in fact, so sympatric speciation of the mixture is in fact 2.5 times more likely than the model of secondary contact and integration. And in Lake Quiloa, this model is 70 times more likely. So coming back at our three main scenarios, the main line of evidence or most support is really for this model of sympatric speciation after admixture and not for the secondary contact integration model. And I'm doing time. Okay, so to sum this up, it seems that population divergence really happened in sympatry. So we can really think Cichlids can still be considered a valid or true case of sympatric speciation, depending on your definition. It seems that several speciation events happened in bursts. So this might be the first empirical example of multi-species outcomes of sympatric speciation. And very interestingly, we find this evidence for these admixture events from the source population into the crater lakes just before they speciate. So this could be evidence that there has been an allopathic origin of genetic variants that later have been recruited in the speciation process to really drive them apart. Maybe you can think of it, maybe the best example for something similar to this is in Raguletis pulmonella flies, where the divergence between the host races somehow, or some of the variants that were involved in, is probably some inversions that came from a population in Mexico. Yeah? So allopathic origin of variants that then drive divergence in sympatry. But I have to stress here that this is of course just a pattern. And we really have now to, first of all, establish with more markers and more data if this pattern is really true of the admixture event. And then, of course, if this really may or may not have facilitated speciation. So we still have a lot of work to do. But it's a quite cool hypothesis, and this really could explain how this might have happened. And yeah, with that, before I close, just please check out our paper. It should be out soon in PLOS Genetics. You can see all the details there. And thank you for your attention, my collaborators here, and the funding sources. Thanks.